Okay, so welcome to week 9, and this is your pre-prac video for this week's prac 2, drugs and allergies, coming up this Thursday. You should find that this prac ties in quite well with the lecture material that you've had so far on inflammation. So a bit of background before we start. Our main defence against foreign pathogens or foreign anything includes the cells of the immune system, of which there are many. Now, as this cartoon, which is grossly oversimplified, indicates, usually an immune response is activated by foreign antigens appearing. They get detected by the cells of the immune system, and they're like a giant red flag to these cells. So, for example, you've got this guy who is covered in antigens that mark him as foreign. And so that very clearly indicates to the cells of the immune system that they need to get rid of this foreign agent with this foreign antigen on it. Now, usually, and this is relevant to your prac for today, antigens are generally proteins, and generally they are markers on the cell surface. So, as the name suggests, they mark the cell as foreign, inducing that immune response. In terms of the immune response, you can think of it in two different phases. The first exposure to any type of antigen will cause what's known as a primary immune response, where antibodies that are quite specific to the foreign antigen that's been detected will be generated. And this is known as the primary immune response, where you have an increase in antibody numbers as they get synthesized, but it may not necessarily be a particularly quick response. The thing about having this primary immune response is that it primes the immune system, and it induces a type of memory in immune cells, so that when you get second exposure to this same antigen at a later point in time, you'll get this much bigger, much amplified secondary immune response, where the generation of antibodies is much greater. You get a lot more of them, and you'll tend to find that the immune response happens quicker. And so overall, it's much more efficient second time round. Occasionally, what you'll have is hypersensitivity, and this is better known as allergy. Usually, this occurs when the immune response is activated in response to something that is actually harmless. And if you think about typical allergies, these might happen in response to things like pollen, mold, dust mites. Think of any allergies you have yourself. The exposure to that allergen or to that foreign antigen has induced an inappropriate activation of your immune cells. So what usually happens in a type 1 hypersensitivity reaction, and this is relevant for today's prac, initial exposure to the allergen, and for this prac, the allergen that's been used has been protein taken from egg albumin, and this is going to be loaded with antigens all over it. What will happen is that exposure to this allergen will result in the production of antibodies. Now, in humans, these are IgE antibodies. In the guinea pig, which is the animal that we've used for this prac, they're actually IgA antibodies. Now, what will happen is that these antibodies will attach themselves to mast cells. And the mast cells contain granules, and they've got a whole lot of very active little mediators inside them. Now, this response, what happens the first time round, is known as sensitization. The problems arise when you get second exposure to the allergen. So in this case, you'll get, for example, egg albumin being injected into the animal. The foreign antigen will be detected by these antibodies that are bound or previously bound themselves to mast cells. Now, upon binding, these mast cells that have been previously sensitized will liberate their contents. They're going to spew out these contents, which include things such as histamine. Histamine is highly reactive, and it's one of a class of mediators known as autocoids. Autocoids include things like histamine, leukotrienes, serotonin or 5-HT, and prostaglandins, which are also relevant to this particular prac. And all of these contribute to those symptoms that we associate with allergy, such as itchiness around a particular area, bronchoconstriction, and nasal congestion. Which brings me to the aim of this particular prac, and that is to demonstrate the involvement of histamine and prostaglandins in a model of type 1 hypersensitivity. So the animal model that we've used, and essentially we've made a guinea pig allergic to egg albumin. And what's happened is that egg albumin, about 3 weeks prior to your prac, egg albumin was injected into a guinea pig. So this would kick off that sensitization reaction, or that sensitization phase. About a week later, this egg albumin was re-injected into the animal. So this would then kick off the allergic reaction. So you can imagine that those of you that have sensitized tissue or tissue from this allergic animal, you're going to have very angry tissue, a whole lot of different autocoids. So looking at the experimental setup, initially you're going to have either control, non-sensitized, or sensitized tissue, which is the guinea pig ileum. All tissue is going to be set up at 34 degrees under 1 gram of tension in Holman's solution, and it's going to be bubbled with carbogen. Now what you need to do at the start of the prac is to figure out another group that you're going to work with on your bench. And what you need to do is, if you're a control group, you need to find yourself a group that has a sensitized tissue, and vice versa. Please don't go and get someone who has the same type of tissue as you. And what you need to do is you need to be fairly well coordinated with your partner group, because at one point fluid needs to get transferred from the sensitized tissue to the control tissue. And you're going to have a look at how this affects the reactivity of your tissue. And specifically, you're going to be looking at the reactivity or the contractile responses of your tissue in response to the drugs acetylcholine and histamine. So again, grossly oversimplified. Here's your ileum, which is just your piece of smooth muscle. Acetylcholine is going to induce contraction via muscarinic receptors, and have a think about what the purpose of acetylcholine is in this particular experiment. Histamine, on the other hand, is also going to induce contractile responses, but via the histamine receptor. Now, for the first of the drugs that we're going to use to investigate the contribution of histamine is going to be meperamine. Meperamine will block the histamine receptor and therefore block contractile responses of the ileum to histamine, whether that is histamine that you've added exogenously, or also the histamine that is being churned out from sensitized tissue as well. The other relevant autocoid, as I mentioned earlier, in allergy is the class of autocoid known as the prostaglandins. Prostaglandins are produced by the enzyme COX from arachidonate, and you can block the production of these prostaglandins by using indomethacin. So by using indomethacin and by using meperamine, we can get a feel for the relative contribution of these autocoids to the response of your tissue in this prac. So the experimental protocol for preparation number 1, which everyone is going to do identically up to this point. So first of all, make sure you know what tissue you have. Is it control? Is it sensitized? The first thing you'll do is test that the tissue is actually viable and contracting properly, and you're going to do that by testing with high-dose acetylcholine. You'll then wash out and obtain 3-point concentration response curves to each of acetylcholine and also histamine. Now you're going to have 3 doses. Now from these, you need to determine which of these gives you a submaximal response. So you don't want the peak, you want one that's kind of in between the largest and the smallest responses you see. So maybe somewhere around 50 to 75%. And you need to test this concentration again for reproducibility. From that point, control groups and sensitised groups are going to follow slightly different protocols. If you're a control, you'll add egg albumin, wait a couple of minutes and wash out, and then you wait. Your partner group will at that point add egg albumin. Now keep in mind what sort of response you would expect to see. As the response stabilises, remove the bathing solution surrounding your tissue, so drain the Holman's solution, and us demonstrators will help you out with that. And you need to transfer this solution to your control partner group. Partner group is going to drain, the control group is going to drain solution from your bath and add the stuff that has been transferred to you from a sensitised tissue. Wait a couple of minutes, see what your tissue does, and wash out. From this point, everyone will come back to a common protocol. You'll repeat responses to acetylcholine and histamine, and then you'll each add egg albumin, leave for a couple of minutes and wash out, observing what responses you get. Preparation number 2 is a little bit more straightforward. You'll get from us fresh sensitised tissue, so everyone's going to have allergic tissue. Wait a couple of minutes for the tissue to stabilise in the bath and determine submaximal responses to acetylcholine and histamine as you did before. And repeat the submaximal response again. Then you're going to add either the histamine blocker mepyramine or the COX inhibitor indomethacin, or both, or some of you will add vehicle. Wait 10 minutes, get your responses to acetylcholine and histamine again, seeing whether they are affected. Wash out, re-add these antagonists and wait. Then add the allergen, the egg albumin, and watch what happens to your response over about 5 minutes. And finally, you'll wash out. So in each case, have a look and see how your responses are being affected. The things to look for. You'll need to measure your responses to acetylcholine and histamine as the change in gram tension from your baseline. And you'll express this as a percentage of your control responses that you get at the start. You'll also be looking at the responses to egg albumin. So egg albumin is going to, in some cases, induce a contractile response, depending on what drugs you have on board. You're going to measure these as also the change in grams from baseline. Note the time taken. How long does it actually take to reach this maximum response? And we've also set up DataPad for you to measure the area under the curve, and this will be in grams per second. And so you can get this area under the curve by pressing Control D. Let us know if you have any hassles with getting this area under the curve. As you're going to be using class data for your prac write-up, make sure that you enter in your own group's data before you go. And let us know if you have any questions about the setup or protocol or the results that you're getting as you go.